Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump in, if you find these deep dives valuable, maybe helps you cut through the noise, please take a second to like, subscribe, and share this with friends or colleagues. It really does support the Sand Science Experience, helps us keep doing this. Okay, so today we're looking at something really fascinating happening right at the synapse, where nerve cells talk to muscles. We're digging into what happens when the mitochondria, the cell's power plants, start to fail. And specifically, using the fruit fly, Drosophila, as a model, new work shows this failure can trigger, well, both helpful signals and total destruction. It all depends on which side of the connection we're talking about.
That's the crux of it, yeah. It's all about context. Neurons, as you know, they need a ton of energy. Most of that energy, ATP, comes from the electron transport chain in the mitochondria, and kicking that whole process off is Mitochondrial Complex I, MCI. It's this huge complex machine, like 42 different parts, absolutely essential. And we know in humans, when complex I doesn't work right, you get really severe neurological problems, things like Leigh syndrome.
All right, serious stuff. And the standard thinking has always been complex I breaks down, it starts leaking. Reactive oxygen species, ROS.
Exactly, ROS, superoxide specifically. And yeah, usually we think of ROS as just damaging, oxidative stress, cellular garbage causing problems.
But this study in flies, at their version of the nerve muscle connection, the NMJ found something. Well, weird.
Extremely puzzling, yeah. So they deliberately messed up complex I, depleted a key part called ND20L just in the motor neurons. And when you look, disaster. The mitochondria look terrible, all clumped up, fragmented.
Almost like they'd lost the proteins that normally help them fuse together like marf.
Precisely. Very similar look. And crucially, there were way fewer mitochondria down at the actual synapse, the axon terminal where the action happens. You can see it in their first figures.
Okay, so visually it's a train wreck. Mitochondria are broken, missing. But here's the kicker, right? The actual communication, the signal from nerve to muscle,
remained perfectly normal. Evoked synaptic excitation was unchanged. Despite the engine room looking like it exploded, the lights stayed on.
which just screams compensation, doesn't it? The system must be doing something actively to keep things working.
Exactly. It implies there's a powerful hidden fix going on, and that led straight to the big idea. Maybe that ROS, that stuff we thought was just toxic waste.
Maybe it's actually the signal triggering the fix.
That's what it looks like. But crucially, it acts as a signal whose meaning, whose effect, is completely different depending on whether it's in the neuron or the muscle.
A real tale of two tissues, separated by just that tiny synaptic gap.
That's a great way to put it. Let's break it down. First, the presynaptic side, the neuron. When complex I fails there, the ROS that builds up, let's call it neuronal ROS or NROS, it acts like a danger signal. It tells the neuron, hey, energy crisis incoming, adapt. And the neuron launches this amazing compensatory process to keep the signal flowing normally to the muscle.
Okay, so NROS is a signal for help, for adaptation.
Yeah.
But then you hop across the synapse to the muscle, the postsynaptic side. What happens if complex I fails there?
Total opposite. When you get muscle ROS, or MROS, it's purely destructive, no adaptation. Instead, you see the synapse start to degenerate, the structure falls apart, communication breaks down, and you see it behaviorally, too. The fly larvae have real trouble crawling.
Wow. Okay, let's dive into that neuron adaptation first. How does it actually compensate? You said it was structural.
Yeah, it beefs up its transmitter release sites. Specifically, they saw this huge increase, about 40%, in a key protein called Brook pilot, BRP.
BRP. That's like the main scaffold at the active zone where the neurotransmitters get released.
Exactly. Think of it like the neuron is installing a bigger, stronger antenna. If the power is weak, make the signal transmission point much more robust to ensure the message gets through loud and clear.
Okay, a bigger antenna makes sense.
Yeah.
But how do we know it's the ROS, the NROS, that's actually triggering this BRP buildup?
Ah, the scavenger experiments. That's the key proof. They went in with tools that specifically mop up mitochondrial ROS superoxide, things like the enzyme SOD2, or a drug called NACA.
So they cleaned up the NRS.
They neutralized it. And as soon as the ROS signal was gone, guess what happened to that big BRP enhancement?
It went away.
Completely reversed, back to normal levels. It's like the cell said, okay, danger's past, we don't need the emergency reinforcement anymore.
And crucially, what happened to the function when they blocked the compensation by removing the ROS?
Then finally you see the underlying problem. With the ROS gone and the BRP structure back to normal, the synaptic signal actually dropped. A small drop, but significant.
proving the ROS really was driving that compensatory BRP structure, which was actively masking the functional defect all along.
Exactly. The NROS wasn't just a side effect. It was the essential signal maintaining function.
Okay, but building that massive BRP structure, that 40% increase, that must cost energy, right? Especially when the main power plant, Complex I, is already failing. Where's the energy coming from?
Excellent question. The neuron needs backup power, and the study found it: glycolysis.
The cell's sort of emergency generator, the non-mitochondrial way to make ATP.
Precisely. They showed that if you block glycolysis using drugs like lanodomine or 2DG, that whole BRP enhancement fails. It can't happen.
So glycolysis steps in to pay the energy bill for building and maintaining that compensatory structure.
It seems so. It fills the energy gap left by the failing Complex I, enabling the adaptation.
Wow. So it's NROS is the signal. Glycolysis is the backup power. But you mentioned calcium earlier. Is that involved too?
It is. Turns out, the failing Complex I makes the synapse really sensitive to problems with calcium handling. The whole compensatory mechanism, this BRP buildup, it only works if calcium levels are managed properly. If you mess with both how mitochondria buffer calcium using the mitochondrial calcium uniporter MCU and how the ER releases its calcium stores through channels like RYR and IP3R.
So if you disrupt calcium from both sources.
Then the compensation crashes, function tanks. So the neuron needs the ROS signal, the glycolytic energy, and stable calcium control to pull off this adaptive rescue. It's quite sophisticated.
Okay, that's the neuron's impressive defense. But now, let's look across the gap to the muscle, where the story is completely different. Same problem. Complex I failure, but disaster strikes.
Right. In the muscle, there's no sign of adaptation. Depleting Complex I there causes an immediate sharp drop in synaptic transmission. The muscle just isn't receiving the signal properly.
And that matches the crawling problems they saw.
Directly correlates, yes. The functional breakdown is immediate.
And structurally, what's the MROS doing to the muscle tissue itself?
It's causing degeneration, basically. You see, the synapse actually shrinks fewer connections, smaller connections, what they call boutons. But the real damage seems to be to the internal structure within the muscle. Right at the receiving end, the postsynaptic density, or PSD, the scaffolding just collapses.
What does that look like? What proteins are affected?
They saw big reductions in key structural proteins, particularly one called discs large, or DL, and another called alpha spectrin.
These are like the main support beams holding the whole receiving apparatus together.
That's a good analogy. They provide the essential structure. And when DL and spectrin levels drop, the PSD becomes disorganized, it loses its integrity.
And if the receiving structure falls apart?
Then the receivers themselves, the glutamate receptors, GLURI-IA and GLURA3 in this case, they get lost or misplaced. This leads to what they call misapposition. The neuron's transmitter release site, that BRP antenna, might still be there, but the muscle's receptors aren't lined up properly underneath it anymore.
So the signal is sent, but there's no one home to receive it, effectively.
Or the receiver is in the wrong place. The communication line is physically broken because the postsynaptic structure has crumbled.
Such a stark contrast. Neuron adapts, muscle degenerates, which brings us back, I guess, to the final proof. The antioxidants again.
Yes, the rescue experiments in the muscle really seal the deal. If MROS is the toxin causing all this destruction, then getting rid of it should fix things.
Right, you did it.
Completely. When they used those same mitochondrial-specific ROS scavengers, SOD2 or NACA in the muscle, everything recovered. NMJ growth went back to normal, the DL spectrin organization was restored, the glutamate receptors lined up properly again, synaptic transmission was fixed, and importantly,
The flies could crawl properly again.
the crawling behavior returned to normal. Cleaning up the mitochondrial ROS in the muscle rescued the structure, the function, and the behavior.
And the specificity was key here too, wasn't it? It had to be mitochondrial ROS.
Absolutely critical. They tried other scavengers, ones that work in the main cell fluid, the cytosol like SOD1, or in other organelles like peroxisomes using catalose. No effect. Didn't rescue anything. It proved the problem wasn't just any ROS anywhere in the cell. It was specifically the superoxide building up inside the mitochondria of the muscle cell that was driving the degeneration.
Okay, let's try and pull this all together then. It sounds like the loss of this crucial energy component, Complex I, causes this ROS buildup.
Mitochondrial ROS.
But what happens next is completely dependent on the context, on the cell type.
Precisely. In the neuron, that NROS acts as a beneficial internal danger signal. It kicks off this complex but effective compensation, boosting the active zone with BRP, fueled by backup glycolysis and reliant on stable calcium, all to keep the synapse firing.
But in the muscle, the same ROS buildup MROS acts like a straight-up toxin.
Right. It triggers degeneration, structural collapse of the postsynaptic side, loss of receptors, functional failure, and ultimately behavioral problems. No adaptation, just destruction.
So this really challenges that simple view of ROS as just bad. It's a signal, a very local one, that can mean different things in different places. Adaptation here, destruction there.
It does. And thinking about human mitochondrial diseases, which often hit both nerves and muscles, well, this work suggests things could be complicated. You know, if you were designing a therapy, maybe an antioxidant treatment, you might need to be careful. Do you want to block the destructive ROS in the muscle, but maybe preserve the potentially adaptive ROS signaling in the neuron?
So a simple systemic mop up all the ROS approach might not be ideal. It could even be counterproductive in some tissues.
It's certainly something this research makes you consider. The context is everything.
Which leaves us with a really interesting thought, doesn't it? If the nervous system can take something like ROS, usually seen as toxic, and repurpose it as a vital adaptive signal, what other molecules, maybe other byproducts or things we currently dismiss as just damage, might actually be playing hidden, crucial signaling roles in keeping our cells, especially our demanding neurons, working?
That's the million-dollar question, isn't it? What else are we missing?
Something for all of us to think about. If you found this dive into cellular adaptation and destruction interesting, please do like, subscribe, and share it. Help spread the science. Thanks for joining us for another edition of the Sand Science Experience.